Okay, hello everyone. So today we are going to take a look at the process of photosynthesis. So we've talked about how we can construct a photovoltaic cell, and we've talked about how the sun possesses this energy. So biologically speaking, I mean, from a biochemistry standpoint, what we want to look at is how is nature already taking this light energy from the sun and using it to create a usable energy source, which we would consider as a sugar. So let's take a look. Okay, a couple different things to look at and think about before we get started. So, what's our goal here is to determine how nature uses the sun's energy to produce a usable form of chemical energy. So, how does it produce something like an ATP molecule? We're not going to look at this in the context of combined with cellular respiration, but we're going to focus primarily on photosynthesis and primarily on the light reactions and how is this creating chemical energy from solar energy. So we are primarily going to focus on the light reactions to make connections in contrast to how the sun's energy is harnessed, to contrast that with the photovoltaic cell. The dark reactions are essential, which we will discuss, but mainly we're going to focus on the light. And then we need to make another side note, which is that there are a few different mechanisms for photosynthesis. This is primarily what we're going to look at, C3 photosynthesis, but there are also a couple other pathways, C4 and CAM. So primarily what we're talking about here is C3 photosynthesis. So just a note to make before we get started. So where is this process happening? Okay. So photosynthetic plants possess organelles that are called chloroplasts. Okay, so this is an image of what we're looking at when we're talking about a chloroplast in general. Okay, so the light reactions are taking place in the thylakoid membranes, okay, and the dark reactions are taking place in the stroma. So if we look at where those are located, just from a location sense, right, the stroma is kind of filling this up, and then we've got these thylakoid membranes, these kind of stacks of thylakoids that are present inside this organelle, okay, and so this is where our light reactions are taking place. All right, so let's look at the light reactions. Okay, there is a lot going on in this image, so let's kind of break this down and kind of focus on a few different things, okay? And we're going to start here, okay, with this H nu. Okay, so remember, when we talk about the energy, okay, energy is equal to H nu, or we can say that energy is equal to hc over lambda when we're talking about a specific wave, okay? So this is our energy, okay, that's coming in the form of light energy, and it's striking the chloroplast, okay? So specifically, what's going to be able to absorb that is chlorophyll molecules, okay? So if we look at an overview of this, we've got the thylakoid membrane, which is where the light reactions are taking place, okay? And inside this thylakoid membrane, we can look at these different structures, okay? So we've got several different structures that we can observe and look at, okay? So we've got photosystem 2, okay? So PS2. We've got photosystem 1, okay, PS1, and then we have this cytochrome B6F complex here. Okay, so when we're looking at these are protein complexes. Okay, so each of these are transmembrane proteins. We can see them spanning the thylakoid membrane, and so they have a specific organization that allows their function to be carried out. Okay, so we're going to take a look at what is going on within these protein complexes of the molecules to allow this to create energy. Okay, so let's kind of take a step back before we look at these protein complexes and talk about this light absorption again. Okay, so remember, we've got light, okay, so energy is equal to hc over lambda in the form of a photon. Okay, so this is our light and our packet of light energy, if you will, and it's going to hit some electron. And if it has the right amount of energy, okay, it can be absorbed by the respective chlorophyll molecules, okay? And these molecules depend on the photosystem, all right? So just like we said, we've got two different photosystems that are in place here, okay? We've got photosystem 2 and we've got photosystem 1. Okay, so when this light is absorbed, if it's got the right energy to excite the electron, then it will excite it to the excited state. Okay, so once it's to the excited state, we talked about how there's two different things that can happen. Okay, either that is going to relax back down to the ground state, and when it relaxes back down to the ground state, then it would emit whatever light, okay, that will be emitted by the molecule. Okay, so it will emit a photon, and that photon will contain some discrete energy value, and it will, if it's in the visible range, then we can detect it as a color. Okay, but simultaneously, so that's one option. So when it does relax, it could emit radiation, and we could call this fluorescence or phosphorescence. Okay, the other option that can happen is it can be transferred to another chlorophyll molecule. Okay, and this is what we're looking for in the photosystem to kind of get this process started. Okay, so once it's excited, it can be transferred to another chlorophyll. Okay, when we're talking about the structure of these chlorophyll molecules, okay, these differ in a functional group that's within what's called the porphyrin ring. Okay, so if we're talking about the porphyrin ring, okay, we're talking about this region of these chlorophyll molecules. Okay, and we can see there's a difference in this functional group here. That is what is different between chlorophyll A and chlorophyll B. Okay, and this porphyrin ring is what allows for the absorption of the light energy. Okay, so when we get this electron transfer, let's look at what's happening in the electron transfer once that electron in the porphyrin ring gets excited. Okay, you need to edit that and go backwards on the porphyrin ring piece. Okay, so go backwards. Okay, so once an electron in the chlorophyll molecule is excited, it can be transferred to other chlorophyll molecules in the reaction center. Okay, so if we take a look at what's going on here, here's our light energy. Okay, so our light energy is being input, and then we've got these chlorophyll molecules. Okay, chlorophyll molecules, and this can transfer the electrons until we reach the reaction center. Okay, remember, the light energy is exciting these electrons, and then they've got a higher energy and have to either relax or they have to move. Okay, and what the photosystem wants is for them to be moved and to transfer to other chlorophyll molecules, ultimately with a transfer to the reaction center. Okay, with a transfer to the reaction center, then we see that the electron can be picked up by the pheophyton. Okay, and once it's here, it can continue to move through the photosystem complexes. Okay, so if we go back to this overview schematic, okay, we see that this electron is excited. It can be transferred to the reaction center, and then it's going to move to the pheophyton, and it's going to start to move through these transmembrane proteins and be transferred to the photosystem, to the cytochrome B6 complex, and to the photosystem 1. So if we see this electron here, okay, we can follow its trajectory along and through these transmembrane proteins, through these different quinone molecules, and on through the cytochrome complex and into the next photosystem. Okay, ultimately with the goal to create chemical storage of this energy, because once it's excited, it's got more energy, and we have to be able to, or the plant has to be able to use that to create an energy source for fuel. Okay, so let's take a closer look at these protein complexes that exist in photosystem 2 and in photosystem 1. Okay, so here's our places where the light is coming in. Remember, H nu is energy. That's our light energy source. Okay, so if we look at these complexes, we can see all of these different proteins that are existing here. Okay, so we could consider this kind of like a super complex, and we've got all these different subunits that compose photosystem 2 and that compose photosystem 1. So each of these play a different role in transferring the electrons and moving them through a redox chain. Okay, so if we remember and think about redox reactions, ultimately what we're looking at are different reduction or oxidation processes that ultimately are talking about the transfer of electrons. Okay, so things are either gaining or losing electrons, which allows the shuttle of these electrons through the system. Okay, so when we're looking, so let's take a look at photosystem 2. Okay, so D1 and D2, this is our reaction center subunits. Okay, there also are different antenna units and different subunits that make this up that interact in what we would consider that are contributing to the light reactions, which are where we evolve oxygen. Okay, so this is where O2 is being produced as a product of these redox reactions. Okay, so kind of as a general overview of this, what I want us to see is that this is a protein kind of like a super complex as we could consider it. And it's not just like this one thing that composes the photosystem 2 or photosystem 1, but we have all these different intricate proteins that are all spanning the membrane in order to function to generate this usable energy source. Okay. So generally speaking, what are we looking at? Okay. And I like this schematic because it kind of shows us what's going on if we're thinking about this if we're thinking about photosynthesis as a way to generate kind of an electrical system and how we can observe the generation of energy through this process. Okay, ultimately this can happen because we're seeing carbon metabolism. Okay, so carbon fixation, where we're taking a CO2 molecule and ultimately what we're creating is a usable carbon source to power or to use as a fuel for organic, like an organism in the context of a plant in this case, which will ultimately fuel ecosystems. Okay, so if we look at this electron transport system and kind of in combination with this legend, here are different wavelengths, okay, that we are seeing enter into the photosystem. Okay, so we've got this visible radiation that can input into the photosystem, and then it can either release visible radiation as well if the excited electron is relaxing, or it could also release some type of heat could be released as well. Okay, we can follow the flow of electrons in this process. Okay, so in photosystem 2, we're getting electrons that are flowing to these plastoquinones, okay, to these different quinone molecules. They're moving into the cytochrome B6 complex. And then we're going through some different oxidation and reduction mechanisms in order to move into photosystem 1. Okay. Then they're going to move through this process again into these different carriers that we're using throughout the process. Okay. So we've got different carriers that are acting in this reduction and oxidation because they are either they're either electron acceptors or donors. Okay. To move these, and what the electron acception and donation does is it creates energy chemical storage. Okay, we've got this moving electron that's at this higher energy state than where we started, and now we can move it through these carriers to ultimately use electron transfer to create usable energy through carbon fixation. Okay, so this light reaction is shuttling these electrons along to interact with this Rubisco enzyme to finally produce some source that we can use. Okay, so let's take a look at what is going to happen in the next step in the dark cycle. Okay, so if we think about this, we've got NADPH and we've got RUBP from the light reactions as a product, and they're going to go to the next cycle in this system. So let's take a look at where those are and how this works. Okay, so here's our NADPH, which is coming from the light reactions, and here's our RUBP. Okay, so RU15BP to be a little bit more specific here. Okay, so what's going to happen is we've got this carbon intake, which we like because that's what we know photosynthesis, we like it because it takes CO2 out of the atmosphere and it gives us oxygen. Okay, and this Rubisco is an enzyme, okay, that is going to interact with the RUBP. Okay, ultimately, what we're ending up with from here is this 3PG molecule. Okay, and this is a precursor to what we're wanting, which is the G3P. Okay, lots of acronyms here, so I apologize for that. But once the RUBP from the light reactions interacts with the Rubisco enzyme, we end up with this precursor, okay? And it goes through a reduction process. Remember, reduction is gain, okay? So NADPH is going to interact here, and it is going to become, once it actively undergoes this reduction, okay, it's going to become NADP plus, which is something that is used in the light reactions. Okay, so we see the NADP plus here. Okay, so this NADP plus will just go right back to the light reactions and be able to function as an electron acceptor there. Okay, so this is going to gain electrons and become this G3P molecule. And ultimately, we want that because it's half of a glucose, okay, which is where we're going to end up with the energy that the plant's going to use to maintain its processes and things like that, okay? So as we kind of think about and look at photosynthesis, we've got a few big takeaways from this, okay? We see a lot of electron movement, okay, through redox reactions, through acceptors, and donors, okay? We've got these protein complexes that are functioning in the transmembrane regions of these proteins, okay? And those are organized in specific ways structurally to allow this process to function. But this is generally how nature is using the light energy from the sun to harvest that light energy and use it to create a source that allows it to sustain itself. Yes, so please let me know if you have any questions, and I hope you have a great day.